We've spent the previous talks understanding how bacteria cause disease. This talk is about turning that understanding into prevention. Vaccines work because we understand pathogenesis. They exploit the infection sequence to prime the immune system before the bacterium arrives. And bacterial vaccines are a fascinating and sometimes underappreciated area of immunology. They don't always work the same way, they don't always target the same thing, and some of them work brilliantly while others are frustratingly limited. Let's understand why using the biology we've already built. This framework is important because it explains something students often find confusing. Why do vaccinated animals sometimes still get colonized or even show mild illness? The answer depends on which step the vaccine targets. A toxoid vaccine, like the one against Clostridium perfringens enterotoxemia, generates antitoxin antibodies. Those antibodies are exquisitely good at neutralizing the toxin before it damages cells. The animal might still have C. perfringens in its gut, the bacterium may still colonize and proliferate in the right conditions, but the toxin is neutralized before it can trigger enterotoxemia. Colonization happens, disease doesn't. That's a clinically successful vaccine, even if it doesn't prevent every step of the infection sequence. Vaccine platform determines its strengths and limitations. Killed bacterians are safe. The bacteria can't replicate, but they often generate weaker cellular immunity because there's no intracellular antigen presentation. They frequently require adjuvants to boost the response and multiple doses to prime adequately. Toxoids are brilliant in their simplicity. If the toxin is the pathogen's primary weapon, neutralize it and you've defeated the infection. The clostridial vaccines used in cattle, horses, sheep, and dogs are mostly toxoids, and they work extraordinarily well. Live attenuated vaccines generally generate the strongest and most durable immunity because the organism replicates briefly and presents antigens in the context of a near natural infection. But they require more careful handling, cold chain management, and carry biosafety considerations. The greatest limitation of bacterial vaccines is antigenic diversity. Tetanus toxin from any strain of C. tetani is essentially identical. There's only one toxin type, so one toxoid generates universal protection. Compare that with E. coli, which has over 180 O antigen types and dozens of H antigen types. A vaccine against one serogroup gives limited or no protection against others. This is why we have strain-specific or serogroup-specific vaccines for some bacteria. It's also why Mycobacterium bovis has been so resistant to effective vaccination. BCG generates reasonable protection in some species, but is inconsistent in cattle, particularly because cell-mediated immunity is required to kill intracellular bacteria, and generating the right type and magnitude of cellular response with a vaccine is much harder than generating antibody. The biology of the pathogen determines the difficulty of the vaccine problem. Individual animal vaccination decisions are made in the context of population-level goals. When you design a pre-calving clostridial booster program for a beef herd, you're not just protecting each cow, you're ensuring that colostral antitoxin antibody is at peak titer at calving, transferring passive immunity to the calf that will carry it for the first weeks of life before the calf's own vaccine responses can be primed. The timing, typically 4 to 6 weeks before calving, is built around maximizing colostral IgG concentration at the time of first milking. That's systems thinking. You're vaccinating the cow to protect the calf via the colostrum, and cold chain failures are devastatingly common. A bacterium left in a truck on a hot summer day for 4 hours may look normal, but have completely denatured protein antigens. The vaccine generates no protective response, but no one knows until the next disease outbreak. Bacterial vaccinology is one of the most exciting frontiers in veterinary medicine right now. The mRNA platform that delivered COVID-19 vaccines to billions of people in record time is being actively applied to bacterial targets. Just imagine an mRNA vaccine that could be rapidly reformulated when a new drug-resistant strain emerges, deployed within weeks rather than years. Reverse vaccinology, using the entire genome sequence of a pathogen to systematically identify every possible surface antigen and screen them computationally for vaccine potential, has already yielded vaccines against Neisseria meningitidis in humans that conventional approaches failed to produce for decades. Transmission blocking vaccines, vaccinating poultry to reduce salmonella shedding, are already in use and represent a model for how animal vaccination programs directly protect food safety. The tools are extraordinary. The conceptual framework is the same one we've used throughout this series. Understand the biology, and you'll know where to intervene. This talk brings the entire series full circle. We started with bacterial cell culture, built a model of pathogenesis, worked through the major pathogen groups, understood resistance, and now we're using all of that knowledge to understand prevention. Vaccines aren't magic, they're applied pathogenesis. Every effective vaccine targets a specific critical step in the infection sequence with a specific immune mechanism. When you understand the biology, the toxin, the capsule, the adhesion, the intracellular niche, you understand why certain vaccines work and why others struggle, and you understand what it will take to develop the next generation of vaccines for the pathogens we haven't solved yet. The biology is always the starting point. Read the question, think about it, we'll then work through it together. This is a question about both vaccine biology and management systems. First, the vaccine. Clostridial vaccines are toxoids. They generate antitoxin antibody specifically against epsilon toxin in type D. For the antitoxin to work, it needs to be present at sufficient titer at the time of toxin exposure. Several things can reduce that titer: inadequate primary vaccination series, only one dose given instead of two, failure to booster annually or semi-annually in high-risk flocks, cold chain failures that degraded the vaccine antigen, or individual variation in immune response. Second, management factors. Enterotoxemia type D, pulpy kidney disease, is precipitated by a sudden dietary change to high carbohydrate feed, which allows C. perfringens to proliferate explosively in the gut and produce epsilon toxin faster than even good antitoxin titers can neutralize. If the precipitating dietary change is severe enough and rapid enough, the toxin production can overwhelm the circulating antibody. This is why dietary management, gradual introduction to grain, is as important as vaccination in enterotoxemia prevention. Vaccines and management are complementary, not alternatives. Read the question, think about it, we'll then work through it together. This is one of the great unsolved questions in veterinary vaccinology, and several hypotheses have been proposed. BCG is a live attenuated strain of M. bovis. It generates cell-mediated immunity, specifically CD4 T cell responses, and macrophage activation, which is exactly what's needed to control intracellular mycobacterial infection. The inconsistency in cattle may relate to several factors: prior sensitization of cattle to environmental mycobacteria, which can block the BCG response, differences in the route and dose of vaccination across studies, genetic variation in cattle immune responses to mycobacterial antigens, and the challenged dose and route used in experimental trials not always reflecting natural exposure. The regulatory complication in cattle is that BCG vaccination causes tuberculin skin test positivity, meaning vaccinated cattle cannot be distinguished from infected cattle using the standard herd test, which underpins national eradication programs. This DIVA problem, differentiating infected from vaccinated animals, is actively being addressed with companion DIVA skin tests using antigens absent from BCG. BCG vaccination of cattle may yet become part of a control strategy, but only once the DIVA problem is solved at a population level. Read the question, think about it. We'll then work through this example together. This is a passive immunity and antigenic match question. Pre-calving vaccination with coliform bacterins works by boosting the dam's antibody titers, specifically against E. coli O antigens and common virulence factors, so that colostrum is enriched with specific IgG. When the calf absorbs colostrum in the first 6 to 12 hours of life, it acquires passive immunity. Several things could have gone wrong. First, failure of passive transfer. Was colostrum fed within 6 hours? Was the calf able to nurse? Was colostral quality adequate? A serum IgG below 10 grams per liter in the calf at 24 to 48 hours confirms failure of passive transfer. Second, antigenic mismatch. E. coli has over 180 O antigen types. The bacterin may not contain the O antigen of the strain causing disease on this farm. Third, maternal vaccine timing. Was the booster given close enough to calving, ideally 4 to 6 weeks prior, to maximize colostral IgG? Fourth, the bacterin itself. Cold chain, correct product. Investigate failure of passive transfer first, as it explains most neonatal septicemia cases, regardless of maternal vaccination status.